Welcome to Genialis. In this video, we'll walk you through our application designed to analyze and visualize data obtained with Roche KAPA RNA HyperPrep kits. We'll show you how you can manage and interpret your RNA sequencing data without having to rely on bioinformatics scripts or collaborators. First, you'll need to upload your data to the platform. This is done on the import data page. The most common way of doing this is by compressed FASTQ files from your computer, but other methods, such as direct import from BaseSpace, FTP, or public databases are available too. Begin by choosing whether your sample is a single or paired end and select your files. Check to make sure the files are paired and matched to the correct samples. Simply drag the files to reorganize them if necessary. All your new samples are automatically put into a collection for easier data maintenance. After the data is uploaded, you'll be redirected to the collection's details page. We recommend processing raw data using the default pipeline developed by bioinformaticians at Genialis and Roche to ensure quality and reproducibility. Choosing an analysis will take you to the Create Analysis page. Usually, all you need to do is select the samples. Advanced users may wish to modify the defaults. Changing a parameter or even the tools can be done with a few simple clicks. Once you're done, click Run Analysis. This will start the process and automatically redirect you back to the collection's details page, where you'll see new data objects and results appear in the samples and collections data cards. Status icons show the analysis progress. Before we continue, let's take a quick look at how data is organized on Genialis expressions. Each time a process or tool is run on your sample, a data object is generated. For example, if you run an alignment process, your reads are aligned to a reference genome and a new object is generated consisting of the aligned reads and other outputs. These new data objects are all linked to your sample. Think of a collection as a folder to organize your samples. You can group samples in a number of ways, such as by experiment or by batch. It's good practice to annotate your data as soon as it's uploaded. Adding metadata to samples can be done in batch by editing a pre-filled template and uploading it back to the app. Alternatively, you can edit the sample's annotation individually. Metadata of a collection should consist of at least a meaningful description, as this field too is indexed for fast search. Once you've uploaded and analyzed your data, you can search for data objects, samples, or collections in the Search and View Results page. Clicking on any of the search results will take you to a detailed view. It's also easy to share a collection and all its samples with your colleagues for seamless collaboration. You can explore RNA sequencing data using interactive visualizations. Let's go ahead and add this collection of RNA-Seq data to the sample basket and navigate to the visualizations page. For the purpose of this tour, we'll be using a dataset of donor-matched, fresh frozen primary breast tumor and adjacent healthy breast tissue obtained from AMS Bio. Duplicate libraries were prepared using either the KAPA RNA HyperPrep kit with riboerase or that of another supplier. We'll begin on the sample comparison tab by asking, are my replicates consistent? And are my experimental treatments and controls distinct from one another? Hierarchical clustering and PCA plot reveal expected clusters of samples, thus validating the experimental design. The next thing we might want to ask is what genes are up and down regulated in the tumor samples compared to controls. In this example, we can see the number of up and down regulated genes. Individual genes of interest can be manually selected from the table or the volcano plot. Selected gene sets can be saved for later, and it follows you around for fast and simple data exploration. For example, you may be interested in visualizing expression patterns. Visual modules immediately refresh with your current selections, and you can visually group your data based on the metadata. It's just as easy to create and fine-tune heat maps and other kinds of plots. In the Gene Sets tab, you can intersect multiple saved gene sets. It's easy to see the number of differentially expressed genes obtained using the two wet lab procedures. About 1,500 genes are uniquely identified by Roche KAPA kits. Selecting a section of the Venn diagram triggers gene ontology enrichment analysis. Further enrichment results are available through a single-click export of your selected genes to Enricher. You can export your results in various formats, including raster, n-vector images, data tables, and figure captions. Send us your questions and comments to support@genialis.com. Thanks for watching.